Hello, everyone. We're glad you've joined us for this webinar, Ion Mobility Mass Spectrometry in the Omics. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Agilent. To learn more about our sponsor, please visit www.agilent.com. Let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question and click Send. To enlarge the slide window, click on the arrows at the top right-hand corner of the presentation window. If you experience technical problems seeing or hearing the presentation, just click on the Support tab found at the top right of the presentation window, or report your problem by typing it into the Ask a Question box located on the far left of your screen. This is an educational webinar and thus offers free continuing education credits. Please click on the Continuing Education's Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Chris Chouinard, the Assistant Professor of Chemistry in the Department of Biomedical and Chemical Engineering and Sciences at the Florida Institute of Technology. To learn more about our speaker, please click on the Speaker tab found at the top right of your screen. Chris, you may now begin your presentation.
Thank you very much for the introduction, Michelle, and I'd also like to start off by thanking Agilent for sponsoring this event. Today, what I'm going to be talking to you about is a tandem technique that's being used increasingly in analytical chemistry, and that is the ion mobility mass spectrometry. Now, many of you may be familiar with the technique of mass spectrometry, but you might not know as much about ion mobility. And so what I'd like to teach you today is some of the ways that we can use this tandem technique to improve our analyses in the omics of proteomics, genomics, and metabolomics. Now, ion mobility spectrometry, although it is a fairly emerging technique in the biological sciences, it's actually a technique that's been around since the 1950s, and it's been used for several decades, primarily in defense purposes. In fact, if you can recall the last time that you went to an airport and had your luggage or your hand swabbed and then analyzed, that instrument was actually an ion mobility spectrometer, now without a mass spectrometer on the back end. But this has been used in other applications like customs houses and ports to test for illicit chemicals, and even has been used in battlefield monitoring. And so how does ion mobility work? Well, like mass spectrometry, it does allow us to separate gas phase ions as they move through some sort of an external field. Now, with the mass spectrometer, we typically use an electric field, although we do have instrumentation that also uses magnetic fields. And in ion mobility, it's no different. We use an electric field to propel ions through some sort of drift region. Now, in its original form, it's still one of the most commonly used types of ion mobility. We have what's called a constant field or drift tube device. And I've included a diagram here where we can actually see that the ions move through this set of electrodes here with a drift gas in place. And as these ions move through under the influence of the electric field, they undergo collisions with this drift gas, such that smaller ions move more quickly than larger ions. Now, there are several other flavors of ion mobility, if you will, that have been developed over the last couple of decades, including one example, which is called field asymmetric ion mobility spectrometry, or FAMES. This is also commonly known as differential mobility spectrometry, or DMS. And in this particular situation, we're actually applying the electrical fields in a perpendicular way compared with the path of the ions through here. In another more recent advent, we have what's called a traveling wave, which is rather than a constant electric field, it's actually a dynamic field where moving waves within the mobility cell actually propel the ions through. And those ions that are larger and less mobile will roll back over the waves, and they'll arrive at the detector at a later time. Now, as I said, within drift tube ion mobility, the simplest form, we have ions pulsed into the drift tube under the influence of a constant electric field. And as they move through, they achieve a constant drift velocity and are able to be separated based on differences in their size, shape, and charge. Smaller ions experience fewer collisions and travel at higher velocity. Now, I'm here in Florida, and for any of you that have traveled to Florida in the summer, you know that it rains every afternoon, and I like to use that as an opportunity to teach my students about the difference between mass spectrometry and ion mobility spectrometry. Imagine that you're walking along campus, of course, with your umbrella, and it starts to rain. Now, you put your umbrella up, and you notice that as you start to walk, the umbrella seems to have a little bit of a restrictive force on you, and that is quite literally a drag as the gas molecules in the air are colliding with the large open structure of the umbrella. But when you close that umbrella and it becomes more compact, you don't have those similar collisions and you're able to walk a little bit faster. Now, if I were to ask you the difference in mass between these two things, the closed and open umbrella, your response would be, well, the mass is the same. And that's exactly what a mass spectrometer gives you, is the ability to very accurately measure the mass of that theoretical umbrella. Whereas with ion mobility, we can actually tell the difference in structure and be able to determine whether that umbrella is open or closed based on how quickly it moves through a drift cell. So what we're actually measuring here is drift time, or the amount of time that it takes for our ions to interact with a detector, such as a mass spectrometer. And we can measure this drift velocity in terms of the length of the drift separation and the drift time that we measure here. We also use a characteristic mobility factor that we call K here, which we calculate based on that drift velocity, as well as the magnitude of the electric field that's used in the separation device. And finally, we typically refer to this in terms of a K0, or a reduced mobility factor, which just takes into account the temperature and the pressure of our separations. Now, this is an important point to note, because just like there are many different kinds of mass spectrometers that can be operated under a wide range of experimental parameters, we have ion mobility instrumentation that will go from low pressure, somewhere in the range of 1 to 4 torr, all the way up to atmospheric pressure. Similarly, we have ion mobility spectrometers that will operate anywhere from ambient temperature all the way down through cryogenic temperatures. Now, for those of you who, like myself, are very visually oriented, I've borrowed this slide from Aaron Baker at NC State University to show a visual representation of how ions with slightly different structure can actually have considerably different mobilities. And here what we have is an example of a couple of fatty acids. And you'll notice in this diagram on the left that they each have one unsaturation, one double bond, but one of them is in the cis configuration and the other is in the trans configuration. Now, it doesn't take any background in chemistry to look at these two molecules and say, well, structurally, they do look different. And indeed, when we pulse them through a mobility drift cell, they'll travel at very different velocities, ultimately arriving at the detector at different times. And this allows us to create a drift time spectrum where they are separated based on those differences in mobility. Now, many of you may be very familiar with other kinds of common analytical techniques, such as chromatography and/or mass spectrometry. Well, one of the ideal things about using ion mobility is that it actually pairs very well on a time scale with both of those techniques that I just mentioned. We like to refer to this as nesting. Now, with chromatography, we're often thinking about separation times on the order of several minutes to even a few hours, depending on the complexity of the sample. So here I have a representative liquid chromatography curve here that's taking place over the course of about 10 minutes. Now, with ion mobility, we're actually able to acquire data on the order of milliseconds, up to tens to low hundreds of milliseconds. And what that means is that for every single LC peak that elutes, we are able to get several ion mobility spectra across that one peak. Now, when we do this with mass spectrometry, we're most commonly doing this with a time-of-flight mass spectrometer. And that's because the time of acquisition for this kind of instrumentation is so short. We're often acquiring full mass spectra on the order of about 100 microseconds. And what this allows us to do, similarly is for every point across an ion mobility spectrum, we are able to get a full mass spectrum and determine what masses are contributing to those ion mobility peaks. Now, what this allows us to do, similarly to the way that we can combine LC with MS and even Tandem MS, allows for significant improvements in signal-to-noise ratio. And I've included a figure from a Graham Cook's article that was actually back in the 1980s, but still holds true. And that's as we increase the number of stages of analysis, going from something like LC or GC to a combination of LC-MS, LC-MS-MS, and now potentially LC with ion mobility and tandem mass spec, we increase the number of stages, thereby reducing our chemical noise considerably, although also slightly reducing our signal. But because of that difference in the decrease between those two, we see an accompanying increase in signal to noise. Now, again, when we couple that to some kind of a mass spectrometer, we see that similar to my previous visual representation, as we pulse ions through a drift tube, they're separated by differences in their size, shape, and charge, and then ultimately, as they approach a time-of-flight mass spectrometer, they're analyzed and they produce a mass spectrum. Now, we can then create these three-dimensional plots of drift time versus mass to charge, and so we can get both sets of information, our ion mobility drift time and our mass to charge value, in one experiment. Now, if we are to couple this with something like tandem mass spec, we're actually able to do this in a global or untargeted fashion whereby we are not isolating any ions in the quadrupole. We're actually just fragmenting those in the collision cell. Many of you will be familiar with this. It's termed all-ion fragmentation. Now, one of the drawbacks to this technique typically is that we're unable to make a good comparison between parent and fragment ions. However, because our ion mobility separation is occurring prior to this fragmentation, what we actually see is that all fragment ions from a given precursor will show up on that exact same drift time line that you see here. And that allows us to easily make these matches between precursor and fragment ions. So what are some of the other advantages to using ion mobility as an analytical technique? Well, because we're getting this additional mode of separation, we can take complex mixtures and potentially be able to get more information and separate compounds that otherwise might coalute chromatographically or might appear at the same mass in a mass spectrum. And so here we have a representative LC retention time plot. And when we look at one of these peaks, this first one, for example, we see that the mass spectrum seems to indicate just one compound there at a mass to charge of 807. But when we look a little bit closer into the ion mobility spectrum for that peak, we actually see that these are two different compounds. And these represent isomers, structurally very similar. These are actually stereoisomers that look exactly the same in both the chromatography and the mass spectrometry domains. But ion mobility gives us the ability to resolve those. Now, with mass spectrometry, we have a mass to charge value, and with chromatography, we have a retention time. In ion mobility, what we like to refer to is collision cross-section. And this can be useful for identification because it allows us to measure these values for known compounds and start to populate libraries, exactly the same way as we do with LC or GC-MS libraries in this case. Now, the way that we get this value is based on a drift time and a number of other experimental parameters that are shown here. Things that include the analyte charge state, the drift tube temperature and pressure, as well as the length of the separation. But when we get these values, we can then use them to have improved confidence in identifying these at a later time. So what does collision cross-section allow us to do? Well, when we make these databases of mass to charge and retention time, we can also throw in collision cross-section and see how that improves our identification. Now we see at the top moving along from left to right, as we start to add these collision cross-sections in for each metabolic feature, we can match not just based on mass to charge, but also on a combination of the two. And that allows us to significantly reduce our false positive rate because we're identifying based on an additional metric. This is essentially four-dimensional data where we have a liquid chromatography or gas chromatography retention time, we have an ion mobility drift time, and then we have a single stage and tandem mass spectrum. What this allows us is increased identification confidence so that we can actually get better information, not just on a chemical composition from high-res accurate mass spectrometry, but also an actual molecule based on our collision cross-sections. Now, because ion mobility is a structure-based separation technique, it allows us to get some structural information. And this is a really huge part of the power of ion mobility, is the ability to look at things like binding strength for complexes, as well as size and shape. And in fact, when ion mobility was first coupled with mass spectrometry back in the 1990s, this was one of the earliest applications, was in looking at biomacromolecules, specifically proteins, and how various changes to things like protein primary structure or different kinds of conditions could change the overall structure or the folding. And you're probably thinking, well, this sounds a lot like what might happen in some kind of neurodegenerative disease where we have these actually misfolding. And indeed, this has been used for decades to study the progression of those diseases and how those proteins are actually folding and unfolding. But let's turn back to the omics at large. And the study of the omics is actually several centuries old. And what this means is we're able to look at the relationship between several different omes within the human body, moving from the genes, which we call the genome, going from genomics down through RNA or the transcriptome, through proteins, the proteome, and finally down to small molecule metabolites or metabolomics. And this metabolome most closely resembles the phenotype, which is what we actually see in the human body. And I particularly like this slide because it also shows how differences in things like our microbiome, as well as environmental exposure, or what we sometimes call the exposome, can also affect the phenotype. Now, looking at things like metabolomics, which is closest to the phenotype, we see that this goes back to ancient times, where actually people would look at things like urine samples and test not only the taste and the smell, as well as the color. So believe it or not, people would actually taste urine and use a wheel like this to try to figure out what kind of disease a sick patient might have. Now, fortunately for us, we have very complex analytical methods now, so we no longer have to taste our samples. Now, one of the cool things about ion mobility and it being a structure-based separation technique is that when we start to plot collision cross-sections for various biomolecules relative to their mass to charge, we start to see these characteristic plots here. And what we can see on the right is that things like lipids in green will have a very different trend line than things like peptides in blue or carbohydrates in yellow. So as we identify some kind of an unknown biomolecule based on both its collision cross-section and its mass to charge, these characteristic trend lines can actually give us more information on what kind of molecule that is. So let's jump into some examples of actual data and look at a few different biomolecular classes and how ion mobility has been used both in my lab as well as in other labs to improve upon those analyses. So my lab does a lot of research with the analysis of steroids. And here we have the synthetic pathway that produces allopregnanolone from the precursor cholesterol. And we see a couple of molecules highlighted here, pregnenolone and 5-alpha-dihydroprogesterone. And if you can see from looking at these two molecules, these are actually structural isomers. So again, they'll look exactly the same in a single stage mass spectrum, and they can be challenging to separate using chromatography. But here, in just under 25 milliseconds, we're actually able to resolve these and measure collision cross-sections that differ considerably at approximately 176 and 191 angstroms squared, respectively. We've used this for a number of different steroid isomer pairs, going from some biologically important compounds like 17-hydroxyprogesterone and 11-deoxycortisol, shown up in the left corner, and corticosterone and 11-deoxycortisol, shown in the top right corner, down through some compounds that are also used in development and doping purposes, like testosterone and epitestosterone, as well as structural isomers androsterone and transandrosterone in the bottom right-hand corner. Now, I mentioned that ion mobility is a separation technique that involves ions colliding with a buffer gas molecule. But I should spend a little bit more time talking about what that buffer gas is. Historically, people have most often used helium as the buffer gas because it most closely resembles a small hard sphere. Now, because of a lot of practical considerations, namely the price and the lack of availability of helium, people have progressed to using nitrogen as a more common buffer gas molecule. But this is one example of an experimental variable that we can change to try to tailor our experiment to better separations. And so here I have an example where we're looking at 3 different isomers: testosterone and its structural isomer dihydroepiandrosterone, as well as its epimer epitestosterone. And we can see that although testosterone is well separated from its 2 isomers, those 2 compounds are almost completely overlapping in the ion mobility domain with nitrogen gas. However, if we were to switch over to something else like a larger, more polarizable drift gas, in this particular case we're using carbon dioxide, we can see that these compounds now have different interactions with the buffer gas molecule, and those differences allow us to start to resolve these compounds. Now, we've seen this similar kind of analysis with other kinds of biomolecules. These are steroid derivatives, bile acids. And we see that with very similar chemistry here, the bottom 2 examples, glycoquinodeoxycholic acid and glycoursodeoxycholic acid, the only difference is in the stereo configuration of a hydroxyl group. And in the top case, glycodeoxycholic acid, we actually have that hydroxyl group in a different position on the C ring instead. And what we can see here is using ultra-high resolution ion mobility, we're able to resolve these fairly well. Let's move on to a different kind of biomolecule in the case of glycans. Now, glycans are a really interesting class here because they have so much differences in their structural diversity of these compounds. We have different sugar monomer isomers, we have different kinds of linkage, and we also have things like differences in 1,6 versus 1,4 and even alpha-beta linkages shown here. Now, these are a very challenging class for mass spectrometry-based separations, and certainly a challenging class for chromatography. But what we can see is differences in these linkages, for example, allow us, as shown here on the right-hand side, these can be separated using ion mobility. Now, I mentioned that we can get some kind of conformational ordering, or these trend lines within an ion mobility drift time versus mass plot. But we also see a couple of other examples where this can be seen for charge state as well. I've told you that ion mobility is a separation based on size, shape, and charge. And what we see is that ions of similar mass but higher charge will travel at a much faster mobility time than we see with those that have a lower charge. And what that allows us to do is separate all of these into characteristic trend lines. We can also separate things based on differences in the lengths of some of their functional groups. Here we have tetraalkyl ammonium salts that go all the way from a 3, that's a propyl group, all the way up through a 16 carbon chain. And these you can see increase not just in mass, but also in their ion mobility drift time. Now, moving on to fatty acids and lipids, which are another great example of a structurally diverse class. Here we're going to expand upon that example that I showed you before with 2 fatty acids differing in the configuration, cis versus trans, of their double bond. Here we actually have an 18:1 fatty acid, meaning that we've got 18 carbons in the chain with 1 unsaturation. We've got 4 different examples here, 2 of them with the carbon double bond at the 9 position and the other 2 at the 11 position. And for each of those, we've got that double bond in the cis versus the trans configuration. And these are then compared with the completely saturated 18:0 fatty acid. And what we see here in these bottom left 2 figures is that we're actually able to get good ion mobility resolution between these based on differences in the position of the double bond position, as well as the configuration being cis or trans. Now, we can also look at building upon fatty acids and moving into lipids. With lipids, we have an additional way that these can differ in their structure, and that's the difference between where the connectivity of those fatty acyl tails is on the head. We can have those in the SN1 or SN2 position, or we can take those tails and switch them. Here on the left-hand example, we have two fatty acyl tails, one with 16 carbons and no unsaturations, and the other with 18 carbons and no unsaturations. And we see in that blue versus black representation, where the position of those is changed relative to SN1 and SN2, that they actually have slightly different mobilities. We can also have differences in their SN backbone attachment, as you can see here, relative to that, phosphate moiety there, where we have those actual fatty acyl tails can change the mobility quite a bit. Now, we've already mentioned the difference between cis and trans orientation, but here we have another example. This is a phosphatidylethanolamine, a PE, with 18 carbons on each chain and one double bond in either the cis configuration shown at the top or the trans configuration shown at the bottom. And what we see here is that these actually differ again in their ion mobility. We can also have stereochemical differences. This one on the right is an example of two ceramides where the hydroxyl is either in the alpha or beta position on that tail. And we can see that, again, this changes their mobility quite a bit. Let's jump into another example of a small molecule, another biomolecule, and this is vitamin D metabolites. And this is a really important case of two structural isomers that are often they're often confused with one another. They're present in much different populations of humans, from infants to pediatrics and even adult populations. But there's a couple of issues between these two different isomers. One of them has a much higher calcium regulation. That's the 25-hydroxy vitamin D3 shown at the top right. And the bottom one has a reduced calcium regulation. It has also been shown to be linked to potentially other diseases. Now, in this case, because they're isomers and because many LC methods do not differentiate them, we actually can get some kind of bias in our measurements. When we're really wanting to quantitate just 25-hydroxy vitamin D3, we can get a positive bias based on the presence of this epimer shown here. So, as I hope you're thinking by now, well, if they're structurally different, can we potentially resolve them using ion mobility? Well, when we look at these compounds in the most common ion that we would analyze, we're looking at the M+H with a water loss at mass to charge 383. And unfortunately, as you can see from the ion mobility spectrum here, they are nearly overlapping. We don't get great separation here. But this is a great opportunity for me to introduce another way that we can improve upon ion mobility resolution. When we talk about mass spectrometry, we're always talking about the analysis of ions. Now, these might be protonated or deprotonated ions, or we might add on something else, like a sodium ion, a phosphate ion, or potentially something else like an alkali metal. Now, why does that matter for ion mobility? Well, with the mass spectrum, what we'll see is simply a difference in the mass, potentially better separation in chromatography, or potentially an improvement in the signal intensity within the mass spectrum. But with ion mobility, when we start to look at different ions, such as in this case, sodiated ions, we start to see an improvement in their resolution. So here I'm about to show an ion mobility plot that includes 25-hydroxy D3 as well as its epimer, and I've included 25-hydroxy vitamin D2. This is the plant-derived source of this molecule. And what we see here is that, well, the resolution is slightly better, but still not great. But being good analytical chemists, we wanted to look into the data a little bit more. And what we found, interestingly enough, was that 25-hydroxy vitamin D3 also has this unique drift peak out here at around 30 milliseconds. Now, interestingly enough, this same peak is not seen for the epimer, and it's also seen at a much later drift time, indicating that it's a much larger structure. But what is that? Well, in trying to figure out exactly what that compound was with a considerably larger cross-section, we wanted to turn first to our mass spectra and make sure that what we were looking at was indeed the same compound or some kind of isomer here. And what we found was that in addition to this drift peak that we have here at 30 milliseconds, we also have a couple of longer drift peaks out between 35 and 40 milliseconds. Now, thankfully, mass spectrometry allows us to answer one of those questions. Those peaks out at C are actually dimers that we can see at mass to charge 823. But the mass spectra between A and B look nearly identical. So what is that new compound that we're identifying at a later drift time? Well, we're going to turn to theoretical modeling, and this is a powerful combination often used with ion mobility to get a better sense for what kind of structural differences are really contributing to potential separation. Now, when we think about ions in mass spec, we're really just making sure that we can indeed ionize compounds to begin with. And so if we add something like a sodium ion on and we see that in our mass spectra, we're pretty happy. And we don't necessarily think about the actual physical placement of that sodium ion within the molecule. But what we see is 25-hydroxy vitamin D3 is essentially a hydrocarbon, save for the presence of a hydroxyl group at each end of the molecule. And so when we add that sodium ion in with its positive charge, it's most likely going to interact with one or the other hydroxyl group based on the partial negative on the oxygen atom. But there's also another potential opportunity here, and that is that based on the structural flexibility of this molecule, we can actually have this wraparound such that both hydroxyl groups are interacting with the sodium ion simultaneously. And what we see here is when we actually do theoretical modeling to calculate the lowest energy conformation of these molecules, we see that indeed they do adopt this closed low energy conformation, and the agreement between our theoretical collision cross-section and our experimental collision cross-section is quite good. However, what we also find is that there's another local minimum, another potentially accessible structure here, an open conformation in which that sodium ion interacts with only one of the hydroxyl groups. Now, if we actually plot out all of our putative structures from our theoretical modeling and plot them against the difference in those two different structures, the closed conformation versus the open conformation, we see that for 25-hydroxy vitamin D3, the difference in energy is quite small, meaning that both of those conformations could be adopted. However, when we look at the same kind of energy diagram for our epimer, we see that that energy diagram is considerably larger. That open conformation is actually not energetically accessible. And so this peak that appears here, labeled B, is our open conformation of 25-hydroxy vitamin D3 that allows us to resolve these two compounds when otherwise we might not be able to. So let's move into one last example of the omics, moving from very small molecule metabolites up through very large molecules like proteomics, and the way that ion mobility can be used to improve upon that. Now, I've shown an example of a protein here, retinol binding protein 1, and if we were to analyze that using mass spectrometry, our approaches would largely fall into two different categories. The first is what we would call top-down proteomics. This is the analysis of intact or native proteins, and it can be very challenging experimentally. It generally requires very high-resolution mass spec and a lot of expertise in this kind of field. But historically, the easier way and the more commonly used way to identify proteins is to use what's called bottom-up proteomics. And what this means is that we actually cleave these proteins using something like a trypsin digest, and we cut this up into smaller peptides that we can then analyze using anything ranging from a very high-resolution mass spectrometer even to a triple quad instrument for targeted applications. And because this has a less of an instrumental burden, it's a more common method. But where can ion mobility fit in for this? Well, let's take an example of a peptide that I've shown here with a mass to charge of 999, and I've highlighted two different serine residues. And that's because serine is a common location where we can see a post-translational modification, or a PTM, called phosphorylation. And we could potentially phosphorylate either of those two serine residues. Now imagine if we have a mixture of those two different peptides. If we were to try to separate them using even ultra-high pressure liquid chromatography, we would see that they unfortunately are co-eluting in that dimension. Because they're also isomers, we can't differentiate between them using mass spectrometry either. But when we analyze that mixture using ultra-high resolution ion mobility, we show here that we have an arrival time distribution with two different peaks. Not quite baseline resolved, but good enough to tell the difference between these two. And when we then run those two compounds as individual standards, we can actually see that we are able to separate these isomers based on the placement of that phosphorylation group, whether it's that serine towards the N-terminus or whether it's at that serine in the middle of that peptide. Now, when we use this to look at something like a global protein digest, we're able to get an increased number of features that we can identify because we're now taking and separating in three different dimensions. First, our chromatographic retention time. Second, our ion mobility drift time. And lastly, our mass spectrometry mass to charge value. And we can see here this cool plot where we're actually showing the number of features that are identified in three dimensions. Now, one final thing that I want to talk about is some of the ways that we can use ion mobility with other cool innovations to actually get some more information about intact proteins. And here we have a method that's called collision-induced unfolding. What this allows us to do is take native proteins that are in their most common folded state and subject them to increased energy. Now, as these proteins increase in energy, we'll actually get a gas phase unfolding of these, changing not their mass, but changing their structure. And when this structure changes, we'll see that as a corresponding increase in drift time. We can then plot that collision voltage, the energy that we're giving to these proteins as they're unfolding, relative to their drift time so that we can see these characteristic unfolding events. And this tells us a lot about the dynamics of the protein folding as well as the overall structure of those proteins. Now, there are a lot of new directions in ion mobility analysis, and this is really where we're moving to improve the way that we use this in the omics. One particular example is things like higher resolution instrumentation. Vendors have introduced things like trapped ion mobility, as well as a more recent advent, structures for lossless ion manipulation, SLIM, in which we're actually able to get considerable increases in resolution between structurally similar compounds. Now, one of the things that we'd like to look for in the future is being able to couple ion mobility with higher resolution mass spectrometers, not just time-of-flight instruments. This might be things like Orbitrap-based platforms or potentially even FTICR platforms. We'd like to see more continuity in measurements and calibration, as well as population of CCS databases across the wide range of vendor options, as well as the different flavors of ion mobility that I mentioned very early on. To be able to take something with a static field, a constant drift tube-based measurement, and compare that CCS to something like a traveling-based measurement, that's a very powerful continuity there. Finally, we'd like to be able to expand upon using other tandem techniques. Tandem MS with all ion fragmentation, even looking at things like spectroscopic techniques where there's work going on with coupling of ion mobility with UV and or IR spectroscopy, as well as being able to do some gas phase chemistry like ion-ion and ion-molecule reactions. Finally, we'd like to look at some ways that ion mobility can improve upon some of our commonly used sampling techniques. These include things like mass spectrometric imaging, as well as ambient ionization, including things like desorption or DESI, electrospray ionization. Potentially other analytical areas, we'd love to hear about other ways that ion mobility might be able to improve upon your analyses, because there's no limit on the kinds of applications that this could be used for. So finally, I'd like to finish by acknowledging some of my past and current coworkers, including those listed here at the University of Florida, those listed here at Pacific Northwest National Lab, Dr. Erin Baker, as well as my group here at Florida Institute of Technology, including my graduate student, Aurora Burkus-Matasevich, and several other students in my group listed here. And with that, I'd like to again thank Agilent for sponsoring this. And at this point, I would be happy to take any questions that you might have.
Thank you, Chris, for your presentation. A quick reminder for our audience on how to submit questions. simply type them into the drop-down box located on the far left of the presentation window labeled Ask a Question and click on the Send button. Chris will answer as many questions as we have time for. All right, first question is, what are some of the major instrumentation advances that can be expected?
Well, I think I hit upon that a little bit in my last slide, and I think really what this is kind of a twofold approach. One is taking ion mobility to higher resolution, so that when we have compounds that are very, very similar in collision cross-section, much like a lot of those examples I showed, where we clearly have two different peaks for two different isomers, but are not quite baseline resolved, what kind of instrumental approaches can we take to improve upon that? And it's really a matter of how high can we get in resolution. Just like we're always pushing the boundaries of how high we can go with resolving power and mass spectrometry, we look to do the same thing with developing higher resolution ion mobility techniques. And like I said, there are a few notable examples of that that are being developed both commercially and also in research labs, such as the Structures for Lossless Ion Manipulations platform that's being developed at Pacific Northwest National Lab. And I think that's going to really allow us to get better separations than we can with more historic instrumentation. The second thing that I would say in terms of instrumental advances is coupling this with techniques that allow us to get a little bit better information about our molecules. And this could be things like other kinds of fragmentation methods, in addition to just collision-induced dissociation. I'm talking about things like electron-based methods, electron capture dissociation and electron transfer dissociation, as well as some light-based methods like UV photodissociation and infrared multiple photodissociation, all of these that give us a little bit more information about the molecules to help us and identify differences in those molecules that are separated with ion mobility.
Okay, next question. How can resolution be improved in the case of diastereomers or even enantiomers?
That's a good question, and I think that's really that illustrates one of the major advantages to using ion mobility is for things like isomers. So with diastereomers, when we have some differences in the stereochemistry, we're largely able to separate a lot of these, and I've shown a number of examples of them throughout the presentation. Now, in order to make those kind of separations better, I think that goes back to the first question that I answered, which is in our ability to get higher resolution measurements so that very, very subtle structural differences between things like diastereomers and epimers can still be resolved with this. Now, enantiomers are kind of an interesting problem because enantiomers, or mirror images, are essentially have the exact same collision cross-section and by themselves cannot be resolved with ion mobility. However, we've published a number of cases in which we can separate those, mostly by interacting them with some other kind of molecule that has chiral structure to it. So we can get these complexes where two different enantiomers will interact with a chiral molecule and can thus be separated using ion mobility. And I think that's really the only way that it can be done, although it's important to note that these approaches are far from universal and can be technically very challenging to develop. So enantiomers are still really what one might consider the holy grail of ion mobility separations, but they can be done.
I would like to once again thank Chris for his presentation. I'd like to thank the audience for joining us today. Any questions we did not have time for today will be addressed by the speaker via the contact information you provided at the time of registration. We would like to thank LabRoots and our sponsor Agilent for underwriting today's educational webcast. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's event. Until next time, goodbye.